Thank you for that kind introduction. My name is David Keire, and myself and Dongmei Liu are going to present an overview of guidance for industry on control of nitrosamine impurities in human drugs. I am the Deputy Director of the Office of Testing and Research in the Office of Pharmaceutical Quality, which is a CDER laboratory. So before we begin talking about nitrosamines, I want to first talk about the definition of pharmaceutical quality. A quality product of any kind consistently meets the expectation of the user, whether it's a computer, a car, or a cell phone. Drugs are no different. Patients expect safe and effective medicine with every dose they take. And so pharmaceutical quality is assuring that every dose is safe and effective, free of contamination and defects. It's what gives patients confidence in their next dose of medicine. So nitrosamines are everywhere. Things like N-nitrosodimethylamine and other nitrosamines are common contaminants found in low amounts in foods, beverages, cosmetics, water, tobacco products, and consumer goods. And you don't really have to believe me. If you go to the scientific literature and the peer-reviewed literature, you'll find many examples. I provided 4 at the bottom of this slide in the fine print that you can peruse at your as you desire. So there's many pathways where nitrosamines can form. One which is really a focus for the current topic is where you have a reactive amine in the presence of nitrite, and that can lead to the formation of N-nitroso compounds. And why is that such a concern? It's a concern because in ICH, as outlined in ICH M7, which is a document which describes the assessment and control of DNA reactive mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk. And in this document, in this guidance, internationally harmonized guidance, they describe a extremely high carcinogenic potency set of compounds known as the cohort of concern, and in that set of compounds are N-nitroso compounds. So for these molecules in the cohort of concern, acceptable intakes would be significantly lower. And in a marketed product, if these were discovered in a marketed product, normally the discovery of another impurity at a low level may not trigger a need for follow-up measures, but with nitrosamines, as they are part of the cohort of concern, the expectation is that it's necessary to control them in the drug. So I thought it would be educational. It has been educational, I guess, over the last 2 years to where we've been dealing with nitrosamine issues in pharmaceuticals. To go over that timeline, to give you an idea of how things have evolved if you're not familiar with the whole story. So in June of 2018, the FDA was informed of the presence of NDMA from a valsartan manufacturer. In July, there was voluntary recalls of valsartan due to the presence of NDMA. In September, a second nitrosamine and a nitrosodiethylamine was detected in previously recalled valsartan products. And subsequently, in October of 2018, irbesartan was recalled due to NDEA. So irbesartan is another sartan class of drug. In November 2018, losartan was recalled due to NDEA, and losartan was a very widely used drug in this class, in this angiotensin receptor blocker class of drugs, also known as sartans. In December of 2018, the FDA posted interim limits for NDMA and NDA in the ARBs. In February, The FDA posted interim limits for NMBA, yet another nitrosamine compound that was discovered in ARBs. And in March, NMBA was detected in losartan. And at that time, there were shortage concerns about losartan, which again was a widely used cardiovascular control drug. And so a temporary drug shortage mitigation action was taken, where the FDA did not object to losartan with NBA below 9.82 parts per million remaining on the market. This is a temporary measure to keep the market supplied with drug. In June of 2019, the FDA became aware of laboratory testing that detected NDMA in ranitidine, a popular antacid drug known by the brand name Zantac. In August of 2019, a general advice letter to ARB applicants and drug master file holders was posted to FDA.gov. Root causes for nitrosamines in ARBs were discussed. In September 2019, ranitidine was recalled due to the presence of NDMA. In December of 2019, NDMA was detected in some metformin products, but at that time, the recalls were not warranted as the amounts were below AI. In December of 2019, 1-methyl-4-nitrosopiperazine, or MNP, was reported in rifampin, a drug used for the control of latent tuberculosis. In January of 2020, nizatidine, which is very structurally similar to ranitidine, there were recalls due to NDMA. And in April of 2020, the FDA requested withdrawal of all ranitidine from the U.S. market because of data that came to light in terms of the stability of the drug. In May of 2020, metformin extended release was recalled to NDMA because levels were found above the AI. In May of 2020, and also that same month, 1-cyclopentyl-4-nitrosopiperazine, or CPNP, was reported in rifapentine, a second drug used for latent tuberculosis. So over this time period, numerous information requests to industry from the FDA and responses from the industry throughout occurred. And so obviously a lot of learning was going on both sides. In addition, there was very good interactions with international regulators. This included, under our established confidentiality agreement, scientific discussions about acceptable intake limits, sharing laboratory methods and results, coordination of joint inspections where appropriate, and sharing of inspectional findings. And really, the global nature of the drug supply chain really makes this kind of interaction essential to any future global drug safety quality investigations. They were very, very helpful in this regard. So the ultimate outcome of this past 2 years is the industry and regulators have learned a lot about what factors lead to the risk of nitrosamine impurities in pharmaceuticals. We don't know everything, but we certainly know more than we did 2 years ago. So the root causes for nitrosamine contamination really fall into three main categories. They can be process related, they can be supply chain related, or they can be stability related. And any one of these separate elements can lead to the unfortunate discovery of nitrosamines in the drug substance or the drug product. Could be, for example, physical chemical properties of the starting materials, intermediates, or the drug substance. Could be specific process conditions that lead to the conjunction of reactive amines with the source of nitrite. It could be impurities or reactions with raw materials in the process steps. And these all fall into the bucket of things that can be, hopefully, if it's a development process, identified in development using this nitrosamine kind of specific lens. And then the second is, if you do discover this, you can that there's this formation, you can develop control strategies to either eliminate or appropriately minimize the presence of these things in the drug substance or drug product. Now, on the supply chain side, it's been documented that there's been examples, at least, of the use of recovered or recycled materials or other intermediates contaminated with nitrosamines can lead to the presence of nitrosamines in drugs where the synthetic process steps have very little risk. So, you could be doing a risk assessment based on your process, but if you don't look at elements in your supply chain that can lead to contamination, you could get a surprise in your product. And finally, there's examples like ranitidine, where the drug substance itself, under the appropriate conditions, seems to break down into NDMA. So that's associated with the stability of the drug substance. There could also be similar risks associated with formulated drug products, where excipients that may contain nitrite could be brought together with the drug, either the drug substance itself or other amine-containing impurities in the drug product that normally wouldn't be considered a risk. So all these things need to be considered. And I think in the next few slides, I'm going to go through a thought process for how one might do a risk assessment for nitrosamines based on these three categories. So if you have some of these amines used used as reagents or catalysts in your process steps, dimethylformamide is a commonly used solvent, for example. This dimethylformamide can break down into dimethylamine when heated, and that is, of course, a secondary amine, which can which is reactive to nitrite and can form NDMA. That's one example, and these are so any one of these, if you have them in your process and the conditions are correct, could lead to this family of nitrosamines shown here at the bottom of the screen. So the kinds of questions that need to be asked, you have to assess the stages of your API or drug product manufacturing. This is where you start. Or you ask yourself some questions. Are there API or formulation steps that have the potential to have reactive amines in the presence of nitroso compounds? Are there reagents, for example, catalysts or solvents, that may have components that could form nitroso compounds as side reactions? An example of that is DMF as a side reaction forming DMA, and then there's other process steps going on where you have the presence of nitrite. What about intermediates or reagents sourced from suppliers? So if the answer to any of those questions leads you to determine that there's risks of nitrosamines, then there's further steps that need to be taken. Is the nitroso compound a known mutagen as per ICH-M7? And if so, can you determine a drug-specific acceptable intake based on maximum daily dose of the drug? Now, that's important because what you need here is an analytical method that can target the nitrosamines in your drug product or drug substance. And you need the AI and the MDD to establish what your analytical target should be. How sensitive does my method have to be? Once you have that target, you can develop and validate an analytical procedure that's sensitive enough to track and trace nitroso compounds in the manufacturing process. You can conduct an assessment of the nitroso formation and purge capacity of the synthetic route. And hopefully this would be helpful to establish control strategies to minimize the presence of nitrosamines in the drug. If you have a step very early in the synthesis that leads to the formation of nitrosamines, perhaps there's sufficient purge capacity in the following steps to eliminate them, or at least minimize them to a safe level. And stability. Is the nitrosamine content stable over time in the API? It's a very simple question. Does the nitrosamine content increase during formulation? This is where this method comes in, where you have a good method that sensitively detects these things in your drug substance or your drug product so that you can monitor and put it on stability. And finally, there should be some consideration: is nitrosamine amount stable under real-world conditions in the drug product? Things like sitting an open bottle sitting in a medicine cabinet in a human bathroom, the conditions the drug might experience in a hot truck in the middle of the summer or being shipped to its ultimate destination. There should be some consideration of those, since particularly some of these drugs seem to be sensitive to heat. So again, those are just examples. Based on these data, an appropriate control strategy can be proposed for regulators, and hopefully assure the safety of the drug. Of course, in spite of all that knowledge gained, there's still a lot more to be learned in this area. There are many challenges. And particularly the root causes for nitrosamine impurities are unclear in many cases. There's some good hypotheses. I've mentioned a few of them, but investigations are still ongoing. There's not one source. It's multifactorial, really. But I think, luckily, there are some common factors that we can focus on and, again, apply this nitrosamine lens to a particular process and a drug product, and hopefully eliminate or control these things to an acceptable level. On the regulatory side, where recalls are warranted, where things unfortunately are found over the AI in a drug product, there's a lot of questions that the agencies have to ask themselves. Is a drug medically necessary? Will a recall lead to a drug shortage? If a recall occurs, are there alternatives that the patient can switch to? These are the kind of risk-benefit analyses that the agencies will apply on a case-by-case basis to make decisions about regulatory discretion in terms of allowable levels being allowed in the drug. On top of this, there's clearly a lot of interest from patients, from healthcare providers, from the media, and other international regulators, as I spoke to, about what we know and what we can share about what we know. And these are ongoing and will continue for a while. And of course, I have a little bias because I work in the labs, but at the base of all this is validated laboratory testing methods available to assess the quality of these drugs that are found to be at risk. And for the FDA sampling and testing of the U.S. drug. So here's a list of the numbers, I guess. Over the past 2 years, the FDA has posted 6 methods for the ARBs, 2 methods for ranitidine, 2 methods for metformin, and recently we shared 1 method for measurements of both MNP and CPNP in rifampin and rifapentine. Now, if these are going to be applied in any industry lab or other lab, the expectation is that the method should be validated by the user if the resulting data is going to be used to support a required quality assessment of the API or drug product, or if the results are used in a regulatory submission. We developed and validated them in the CDER lab. Hopefully that would speed development and make the validation easier in the industry lab. I would also point you to, there's a recent USP chapter, 1469, on nitrosamine impurities, which is out for comment, that contains 4 methods. And USP has also made 6 reference standards available. And I provide a link to the USPPF that's out. So key things for the detection the suitable analytical procedure measurement performance for trace amounts in the part per million or part per billion of nitrosamines is the application of appropriate measurement technology focused on not only on the drug product or drug substance, but solvents and intermediates across finished dosage forms. And LOQs should be appropriately below AI, and that's touched upon in the guidance. They have to be sufficiently sensitive to establish absence or amounts when you do have them present. And again, we've tried to make analytical procedures developed by the FDA labs available to industry or other people that want to apply them, and hopefully they're helpful in terms of speeding the screening of processes and products for nitrosamines. Yeah, on this slide, I provide some links to some of the ARB and metformin and ranitidine announcements that are been posted on FDA.gov. There's a wealth of information there. There's also the methods are available through these links in case you have an interest of looking at those methods. So, and also on these FDA.gov pages, there are some Q&A that I think are also very useful. I recommend them. One of those is what industry should know about nitrosamine impurities. And in the answer to that is that for the FDA expectations of industry is that manufacturers are responsible for understanding their processes, which include preventing the presence of unacceptable impurities. Manufacturers are also responsible for developing and using suitable methods to detect and limit unacceptable impurities, including any new impurities that may arise when they make changes to their manufacturing processes. And also to answer the question, an answer to the question, can we trust FDA and its approvals and drug surveillance? So we try to maintain with U.S. consumers that we're a trusted source of information about the quality of their drug products. And the FDA is committed to ensuring that medicines Americans take are safe and effective. When we identify drug quality lapses that pose potential risk for patients, we make every effort to understand the issues and provide our best recommendations to the public as quickly and accurately as possible. And we will continue to investigate and work to ensure these types of impurities do not exceed acceptable limits so the patients can continue to take their medicines without concern. With that, I will stop and I will hand over the microphone to Dongmei Liu for more information on the guidance. I thank you for your attention.
Good morning, everyone. My name is Dongmei Liu. I'm from OPPQ CDER of FDA. So in the past session, David provided very nice information on the nitrosamine incidence in US during the past 2 years. The possible root causes of nitrosamine impurities, factors need to be considered in the risk assessment, and the suitable analytical method for nitrosamines. So following that, I'm going to discuss in details on the FDA guidance information with more focus on FDA recommendations on how to control nitrosamine impurities in human drugs. As you all know, FDA recently published the guidance on nitrosamines. It was on September 1st. For your convenience access, the guidance link is included in this slide. This guidance is published as a final guidance, but it is still open to public for comments. The link to the comment submission is also included in this slide. You're welcome to provide comments on this guidance. The topics I'm going to cover here are background, possible root causes of nitrosamines, FDA recommendations to manufacturers that include API manufacturers and drug products manufacturers respectively, and finally, reporting changes to FDA. Some background information. What are nitrosamines? As David have discussed, nitrosamine is a class of compounds having the special feature in its chemical structure. A nitrosyl group bonded to an amine, as shown in this figure. The amines could be secondary, tertiary, or quaternary amines. They can interact with a nitrite salt under acidic conditions. The nitrosamines have been categorized as a cohort of concern compounds by ICH M7 guidance. Besides that, nitrosamine compounds are potent genotoxic agents in several animal species, and some of them are classified as probable or possible human carcinogens by the International Agency for Cancer Research. At that time, based on the common amines used and the common synthetic pathways for APIs, FDA have identified 7 nitrosamine impurities that theoretically could be present in drug products. Actually, 5 of them are already found in APIs and drug products. Currently, more nitrosamine types are discovered and under investigation. Since 2018, the NDMA was discovered in valsartan. FDA has found out numerous batches in several drug products have an acceptable level of nitrosamines. Drug products manufacturers voluntarily recall the affected batches, which lead to drug shortages in some certain drug products, including valsartan, olmesartan, aprosartan, and nizatidine. In April of this year, FDA requested all products of ranitidine withdrawn from the market. FDA also recommended voluntary recalls on the affected batches of some sartans and extended release metformin from some certain manufacturers. All the information is telling us that the nitrosamine issue has huge impact in the pharmaceutical world. Some drug supplies are interrupted, and consequently, the patients who depend on these products suffer from the drug shortages. Although nitrosamine impurities have been found only in some certain drug products, we start to have questions. Is it possible that these nitrosamines may exist in other APIs and drug products? If yes, what are possible reasons? How to do the risk assessment, and what are the steps to take to ensure the quality of the product? So there is urgent need from API and drug product manufacturers on instructions on how to assess the potential nitrosamine issues in the drug products, and what are the prevention or mitigation strategies. Since the initial incident took place, FDA started to take actions to investigate this issue intensively. FDA communicates with public via announcements timely to update the status of drug products, drug shortages, recall information, and provide advice to patients treated with these related medications. Meanwhile, FDA worked closely with industry to remove affected products from the market to ensure public safety. FDA communicated with industries using general advice letters and letters to individual applicants to share up-to-date knowledge and recommendations to industry. Because the nitrosamine issue extends beyond the U.S. drug supply, FDA and other international regulators have partnered to share information, coordinate inspection efforts, communicate on analytical methods, and to develop rapid solutions to ensure the safety and the quality of the drug supply. Due to the urgent needs from industry, development of guidance for industry in this perspective is as high priority. The guidance was already published, and what is covered in this guidance? There are 4 key elements in this guidance. First is root causes. The root causes include those in APIs and drug products other than the API contaminations. The second key element is recommendations. That include acceptable intake limit, recommendations to API manufacturers and drug products manufacturers respectively, also include recommendations on the three-step mitigation strategy. The fourth element is reporting changes to FDA. That includes the timelines for three-step strategy, and this applies to the applications at various status, including approved marketed drug products and pending applications. Root causes. David already explained in very good details, and I'm not going to repeat here. But I want to indicate that some hidden factors, such as contaminated solvents or materials, they are not easily to tell obviously from API route of synthesis. Therefore, some drug products using the APIs, which are manufactured by certain low-risk processes, still were found to be contaminated. In addition to that, the multiple root causes listed above can occur within the same process for API or final drug product. Therefore, multiple approaches may be necessary to identify all potential sources of contamination. Furthermore, each failure mode could result in different nitrosamines in different amounts across batches from the same process and the same API or drug products manufacturers, with contamination detected in some batches but not all. Next, let's step into the recommendations. The first recommendation is the acceptable intake limit. FDA recommended acceptable intake limits for 6 nitrosamines in drug products as indicated in the Table 1. The values are 96 or 26.5 nanogram per day. The smaller the value, the more toxic it is. The AI limit is the daily exposure to compounds that approximate 1 over 100,000 cancer risk after 70 years of exposure. According to toxicology, it is not expected that a nitrosamine impurity will cause cancer when exposure is at or below the acceptable level. The conversion to part per million, the ppm, is based on maximum daily dose. It can be calculated using the AI limit divided by maximum daily dose. So the next question is how to use this AI limit. If there is only one nitrosamine in the drug product and this nitrosamine is listed in this table, then please use the corresponding individual limit. If a nitrosamine is found but not listed in this table, the manufacturers need to contact FDA, and they can use approaches in ICH M7 guidance to determine risk. In the guidance Appendix B, there is also description on the approaches to determine the AI limit for nitrosamines. This also derived from M7 guidance. If there are more than one nitrosamines in the drug products, the total quantity of nitrosamines should be no more than the AI limit of the most potent nitrosamine. Currently, it is 26.5 nanogram per day. Otherwise, please contact FDA. Nitrosamine in drug products are in a very small amount, but they are very toxic. Therefore, there is a requirement for the analytical method for nitrosamines. The limit of quantitation, LOQ, should be no more than 0.03 ppm. If MDD, the maximum daily dose, is high, for example, is more than 1 gram, the LOD and LOQ should be as low as reasonably practical. For APIs, the API manufacturers should control the nitrosamine level to ensure drug products meet this criteria. Please note that the maximum daily dose are likely different from different drug products even using the same API. Therefore, the ppm level in these drug products may vary. FDA recommends a 3-step for mitigation strategy. That includes risk assessment, confirmatory testing if there any risk is identified, and reporting changes. I'm going to provide more details for these steps. The first step is a risk assessment. The manufacturers need to assess the risk of nitrosamine impurities in APIs, marketed products, and products under approved and pending applications. The risk assessment should be conducted in a timely manner based on the prioritization of drugs. Several factors could be considered for this prioritization: the maximum daily dose, treatment duration, therapeutic indication, numbers of patients treated. FDA may recommend certain drug products as higher priority based on the understanding. And here I want to point out that if there is no risk is identified, no further action is needed. The second step is confirmatory testing. The confirmatory testing should be conducted as soon as possible if the risk is identified, and should begin immediately for products considered as high risk. Due to nitrosamines' physiochemical properties, such as low molecular weights, some volatility, and high toxicity, very specific and highly sensitive analytical methods are needed for this purpose. The third step is reporting changes. The manufacturers need to report changes implemented to prevent or reduce nitrosamines. This includes DMF amendments and changes to approved and pending applications. Here, I want to draw your attention that all risk assessment reports should stay at the manufacturer site, and they should be available upon request. Recommendation to the API manufacturers. These recommendations are corresponding to the root causes of API contaminations. First recommendation: API manufacturers should optimize the design of the manufacturing process during rotosynthesis development. Several factors should be considered. First, avoid reaction conditions that may produce nitrosamines whenever possible. Second, when use secondary, tertiary, or quaternary amines, be aware of nitrosiding agents. Third, use caution when the rotosynthesis involves the use of amide solvents, as they may contain amine impurities, and some process conditions may cause degradation to form secondary amines. Fourth, replace nitrites with other quenching agents for azide decomposition processes if possible. Fifth, control the reaction conditions such as pH, temperature, and reaction time, or the sequences of reactions, such as consider removing quenching steps from the main reaction mixture, and ensure no carryover of amines and nitrosiding agents into subsequent steps. The second recommendation is that the API manufacturers should audit their supply chains and monitor them for any at-risk materials. API manufacturers should maintain records including the name of the raw material manufacturers, suppliers, any repackers, and distributors who handle the material before the API manufacturer. The third recommendation: in order to avoid cross-contamination when recovered materials are used, the recovered materials should meet appropriate standards before reuse. If it is outsourced to third-party contractors, the API manufacturer should audit the contractor's validation of cleaning procedures. If nitrosamines are found in the API batches, what should API manufacturers do? There are two scenarios. First one, if the nitrosamine level is above LOQ but no more than the AI limit, the manufacturer should develop control strategy, including specification limits, to ensure the nitrosamine levels are below AI limit. Given existing uncertainties regarding nitrosamine impurities and their presence in the drug products, for any at-risk APIs with an impurity detected above the LOQ, testing of each batch on release should be conducted. Alternate approaches, such as upstream test of an intermediate, should be supported by sufficient process understanding, and evidence of adequate control should be submitted to FDA in a supplement prior to implementation. The second scenario is, if the nitrosamine level is above the AI limit, the batches should not be released by the API manufacturer for distribution, unless, with a prior FDA agreement, the API is needed to prevent or mitigate a drug shortage. The recommendations to drug products manufacturers are corresponding to the root causes in drug products. The manufacturers should conduct a risk assessment to determine the potential risk. This risk assessment should involve collaboration with the API manufacturers in the identification of the API route of synthesis or other process conditions that put the drug product at risk. Drug products manufacturers should continue testing API lots until they have verified that the API suppliers can consistently manufacture API without an acceptable level of nitrosamine. The second recommendation is that the drug products manufacturers should evaluate whether nitrites could be present during manufacture processes where at-risk APIs are used. The risk assessment should also include evaluation of any pathway, including degradation, that may introduce nitrosamines during drug products manufacture or storage. If nitrosamines are found in the drug products, what should drug products manufacturers do? There are also two scenarios. First, if the nitrosamine levels are above LOQ but no more than the AI limit, the manufacturers should develop a control strategy to ensure the nitrosamine levels are within the AI limit. The control strategy includes specification limits. Such a control strategy is also recommended when the introduction of nitrosamine is inherent due to the API structure, the API route of synthesis, or the manufacture process of the API or drug product. The second scenario, if the nitrosamine levels are above AI limit, product batches should not be released or distributed. If the batches are already in distribution, the manufacturer should contact the FDA so the agency can determine the regulatory action. The manufacturer should contact the agency if a recall is initiated. FDA may exercise regulatory discretion when warranted to prevent or mitigate drug shortages. For the three-step mitigation strategy, FDA recommends different implementation timelines depending on the regulatory status of the drug products. For the approved and marketed drug products, the manufacturer should conclude a risk assessment within 6 months of the publication of this guidance. That means it should be completed by March 1, 2021. The confirmatory testing should start as soon as a risk is identified and should begin immediately for products considered as high risk. Confirmatory testing and the submission of the changes should be concluded within 3 years of this guidance publication. That means the last 2 steps should be completed by September 1, 2023. FDA acknowledged that the implementation timeline includes investigating root causes, identifying changes, and confirming that any proposed changes will minimize the risk. The timelines also include activities conducted by API manufacturers to support the drug products in which they are used. FDA may request an expedited risk assessment, confirmatory testing, or other regulatory action based on the information available to the agency. For the pending applications, FDA also has requirements. For applications at the pre-submission stage, the applicant should conduct a risk assessment in APIs and the proposed drug products, and conduct confirmatory testing if needed prior to the submission of an original application. When necessary, changes to the DMF or application may be submitted in an amendment if they are not included in the original submission filing. Such amendment should be submitted as quickly as possible to minimize any potential adverse impact on the application assessment timeline. For the application at the status of pending with agency, the manufacturers also need to conduct risk assessment and the confirmatory testing if there's any risk. If the nitrosamine levels is more than AI limit, the applicants need to report to FDA. If the nitrosamine level is above LOQ, but no more than the AI limit, the applicants should submit amendment with appropriate control strategies. As a summary of our presentations, in this recently published guidance, it describes possible root causes of nitrosamines impurities in APIs and drug products. It established acceptable intake limits for 6 nitrosamines. It also provides the recommendations on 3 steps to take for prevention or mitigation of the nitrosamine issues. It set timelines for the 3-step mitigation strategy. And lastly, it also includes expectations on the reporting changes to FDA in DMFs and applications at various stages. Lastly, I also want to emphasize that, as we have mentioned earlier, FDA has been coordinating with other international regulators on nitrosamine issues, and there come great agreements on key elements that include systemic approaches of a 3-step mitigation strategy, timeline for implementation of a 3-step strategy. This is the same as those in EMA. Acceptable intake limits for nitrosamines, analytical method and criteria. This harmonization has great benefit, especially for those manufacturers who support APIs and drug products across different countries. Then those manufacturers and applicants can evaluate and address risks and control the products in a consistent approach globally. With the assistance of this guidance, the agency and the manufacturers and applicants will continue to work closely to resolve this nitrosamine issue and ensure safe, effective, and high-quality drug products for the American public. Acknowledgement. David and I want to take this opportunity to thank our colleagues in Office of Pharmaceutical Quality, Office of New Drugs, Office of Generic Drugs, and Office of Regulatory Affairs for their support and valuable input on nitrosamine-related work. We also thank 2 work groups. First one is the OPPQ nitrosamine work group, and the second one is the CDER Task Force work group for their extensive discussions, suggestions, and coordinations. We acknowledge the partnership with EMA, EDQM, OMCL, and cooperation with other international regulators, Singapore HSA, Australia TGA, and Health Canada. Thank you so much for your attention. Now we will take a brief break and take a look at the questions you submitted. We will come back with some answers.
Thank you for those great presentations. If you haven't had a chance to enter your questions into the Q&A pod, please do so now. We're going to take a very short, brief break while we take a look at the questions that we've received, and we'll be right back to start the Q&A session. Welcome back. Thank you for those questions. Please keep sending the questions as we start answering them. Our first question is addressed to Deborah. Is the 6-month deadline for risk assessment, does that also apply to non-marketed products?
That's an excellent question. I'm actually going to send that over to Dongmei, as my expertise is more in the CMC, and Dongmei is more on the guidance type. Thank you.
Hello. Yes. If it's not being marketed, then no. But that should be evaluated before marketing and resumes after the 6-month period.
Thank you for responding to that question. Our next question is addressed to Andre. Is testing mandatory for products even if paper assessment doesn't indicate any risk in terms of nitrosamines?
Yeah, so basically what the applicants need to do is to do a risk assessment at their facility. And if the risk assessment shows that there's really no risk, then no further testing is needed, and you don't need to amend your application. But if there is a risk, and this should be scientifically, evaluated very carefully, and this is confirmed by testing, yes, then this should be some element of a control should be incorporated into your control strategy.
Thank you for responding to that question. Our next question is addressed to Deborah. If nitrosamine impurity is detected below LOQ, is there any action or control strategy required?
That's a good question. After doing the risk assessment, if there is a risk of a nitrosamine being formed and testing actually detects a nitrosamine, even if it's below the LOQ, what that's telling you is a nitrosamine is forming, and some type of control strategy may be necessary. Please remember that testing the nitrosamine in the drug substance is only one part of a control strategy. There's also the option to test for nitrosamines upstream where they're actually formed, and providing a spike purge data to show that they're adequately reduced by the API. There's also the option to control the precursors to help prevent the formation of nitrosamines. However, if you are actually detecting a nitrosamine in your product, that means something is forming, and there should be some discussion and potentially a control strategy added if necessary.
Thank you for responding to that question. Our next question is to Dongmei. NDA and ANDA holders and sponsors and DMF holders should work with contract manufacturers to take the steps in the guidance. However, when the relationship between the API manufacturers, and there's DMF holders in parentheses, and the NDA, ANDA sponsors holders, the lack of transparency between the parties regarding confidential details of manufacturing and process and material specifications may hinder detailed and fruitful discussions and investigations, especially when nitrosamines first occur in the formulation process of the formulated product. Can you please clarify how API industry should bridge this gap?
All right. Yeah, so basically, the risk assessment, that is really needs a collaboration between the API manufacturer and the drug products manufacturers. And so basically, they should share the information as needed, as much as possible, and to ensure the final product quality. So please also see ICH Q10, the Pharmaceutical Quality System Guidance, and for more information. Yet collaboration definitely that is needed. Is strongly encouraged. Thank you.
Thank you for responding to that question, and we have another question for Dongmei. The guidance doesn't specifically mention biologics. Are they excluded, or might they fall under, or drug products at risk, and it's in quotes, due to other factors described in this guidance?
Okay, so for this guidance, actually, it applies to the chemically synthesized drug products, which include any drug or API or drug products with a chemically synthesized structure. So if there's a biological product, it has the chemical synthesized fragments, then this guidance would be applicable to that, and also to the products where the manufacture process is at risk to the nitrosamine contamination due to other factors. So this guidance doesn't include the biologics that are not related to any chemically synthesized fragments or all the structures.
Thank you for responding to that question. This question is addressed to Tim. The specific nitrosamines listed in the new FDA guidance are slightly different than the specific nitrosamines listed in the EMA and USP guidances. How did the FDA select these specific nitrosamines, and are these the nitrosamines companies will be expected to test for in the future?
Thank you. I can address at least part of that question. The nitrosamines listed in the FDA guidance were evaluated based on the reporting to the agency and identification in various drug substances and drug products. So those are the ones that we were asked to address, or that felt the need to address as we have moved forward in this approach. Additional impurities are identified as we've been moving forward, so we are not completely in lockstep with other agencies or USP in terms of our inclusion of compounds in the guidance documents. Colleagues from our product quality office may want to address this as well.
This is David. I think I can just add that so as the nitrosamines were discovered in drugs, clearly we had our Pharm Tox colleagues take a look at them and establish limits for each one of them in turn. At the same time, we were proactively looking at synthetic processes for drugs that were at risk and identifying other amines that could potentially lead to the formation of nitrosamines under certain conditions. And so that's how this list of nitrosamines has evolved. And right now there are 10 nitrosamines that the FDA labs can test for.
Thank you for responding to that question. We have another question for Tim. Some drugs were taken for very limited time, such as ranitidine injectable. My question is, why this period was not taken into consideration?
Yeah, I would say certainly ICH Guidance M7 addresses the issue of less than lifetime use, and we did take that into consideration in terms of identifying the acceptable intakes for nitrosamines as well. However, given that these compounds belong to that cohort of concern classification that David mentioned earlier, we decided that it was not appropriate at this time anyway to apply a less than lifetime approach. That being said, we are able to implement flexibility in that approach when a particular situation warrants it, especially in cases that might impact the availability of medically necessary drug products.
Thank you for responding to that question. Our next question is to Dongmei. Can the packaging process produce contamination of nitrosamines?
Yes, so actually this information it is from Europe and from some other country. And some certain packaging material, especially for this nitrocellulose in the leading foil, that has been reported to interact with the ink components during the printing. So, but for this in the U.S. market, and there is no influence in the U.S. market drug product yet. So try to, for the packaging material, and try to avoid the nitrocellulose packaging. And so far, it seems the polyester and acrylate material they seems fine. And okay, there's really limited nitrosamine has been in the blister packaging, and so but you know, in this perspective, I should say that, and you know, pay attention to that. But so far, it is not a big impact yet.
Thank you for responding to that question. We have another question for Dongmei. It deals with how to control nitrosamine impurities from recovered solvents outsourced. Should we keep control in APIs or recover the solvents?
In order to avoid the cross contamination, so when the recovered material used, you know, such as you know, solvents or reagent or catalyst, and that may be, you know, that is used during the manufacture process and gets recovered. And so if this has to be used, then recovered material that should be used only in the same step or some step earlier, you know, than the material you get from. So, but for the recovered material, they should meet appropriate standard before reuse. And in addition to that, because we know a lot of recovered material that were actually they were recovered by the third-party contractors, and this kind of situation, the API manufacturers, you know, they should provide specific information about the recovered materials or what's possibly in there, and provide this information to the third-party contractor. Then they can, you know, based on their routine recovered procedure, they may have some kind of modification. At the same time, the API manufacturers should audit the contractor's validation of this cleaning procedure.
Thanks for responding to that question. Just a reminder, if you haven't had a chance to enter your questions into the Q&A pod, please do so right now. Our next question is addressed to Deborah. Do primary amines not give rise to nitrosamines?
Okay, good question. So if you remember your basic organic chemistry, typically a primary amine will react with nitrous acid to form an azo compound. It's the secondary and the tertiary amines that we are more concerned with because both of those can react to form nitrosamines. So if your material, if your compounds, your intermediates, your drug substance contain secondary or tertiary amines, or some of your raw materials are secondary or tertiary amines, then you need to worry about nitrosating agents. And I might just say that we don't want to just focus on nitrous acid. We know that tends to be the one that's most common. But nitrite can be in the process through things such as sodium azide. It can also exist in nitrate. There are other sources of nitrosating agents, such as NOx. So when evaluating your process and you have amines of any type, you know, of secondary or tertiary amines, please take a look at your process holistically and not just focus on nitrite, but look and see if there are other potential sources of nitrosating agents.
Thank you for responding to that question. Our next question is addressed to David. Have you published validated analytical methods?
Yes, so we've supplied a number of methods on FDA.gov. I think if you look in the slides, there's some links, and they'll lead you to where the methods are publicly available. In addition, there's one manuscript that was published in June in APPS-J about testing of metformin products. So hopefully those will be useful resources for people developing methods.
Thank you for responding to that question. Our next question is addressed to Tim and Andre, so Tim will come on first and then pass over to Andre. And it speaks to how to control other nitrosamines such as piperazine nitrosamine from the reported nitrosamine limits.
Thank you. Yeah, and this question is really more general. How do we identify an acceptable intake for a compound which has not already either been listed in FDA or other guidance documents, or even compounds for which there is insufficient carcinogenicity data? So the ICH M7 guidance, again, has recommendations for how to go through this process of identifying an acceptable intake level. Oftentimes it's evaluating the literature regarding the mutagenicity data as well as carcinogenicity data. And certainly if there is a new molecule for which an acceptable intake has not been identified via the FDA guidance or other regulatory guidance document, a calculation based on, oftentimes using a TD50 approach can be used to identify an appropriate acceptable intake level. For compounds where there's inadequate data available to support a compound-specific acceptable intake level, then a read-across approach or using similar compounds for which there is adequate data can be applied. And that approach, if that's the approach a particular application is going down, that justification can be put together and submitted to FDA for evaluation and consideration. I think a number of the compounds listed in the FDA guidance use that approach. If in general we deem that there is no inadequate data to do a compound-specific assessment, then we have at times referred back to NDMA and NDEA to base that acceptable intake level. Andre?
No, Tim, I think you answered the question. I think there's nothing else to add. What else?
Perfect. Thank you for responding to that question. Our next question is addressed to Andre first, and then David can follow up after Andre passes over to David. If you've identified a secondary, tertiary, or quaternary mean in your process or from suppliers, but no nitric acid or salt was identified from any of the sources, such as the supply chain, process, or stability, is your product at risk of nitrosamines impurities?
I think that so basically, yes, of course, secondary and tertiary amines in conjunction with nitrites, are definitely a risk factor. But I think Deborah mentioned that there are other. It's not, that those are not the sole sources of nitrites. Nitric acid, haso, nitrous acid can also be a source. So as you do your risk assessment, it is definitely in a higher risk factor, but I don't think the risk assessment should be solely focused on those on the nitrite source, because we know that there are other means of generating nitrosamine, so those should also be considered. But of course, that should be considered in the context of your risk assessment.
And I'll just add that the test methods are there. So if there is this question that comes up and there's doubts, right? That means that there's uncertainty and potential risk there. Then you might want to apply those available methods to do a screen and give yourself some more information to act upon.
Thank you for responding to that question. Our next question is addressed to Dongmei. Are there guides or recommendations which nitrosamines need to be or should be analyzed and tracked, because it's an unbelievable effort to investigate all species of nitrosamines and several hundred or even several thousands of APIs or drug products?
All right.
So the company should base on their amine used in their route of synthesis in the manufacture process and the synthetic pathways to investigate whether there is something related to these amine structures, and that could form the possible nitrosamines. That is really based on what they have been used in their process, in their route of synthesis. For some exogenous factors such as recovered solvent, it is really hard to predict. But still try to look into something actually possible, and especially something that's already identified in the drug product. And let me see. Okay, I think that's all. Yes. Thank you.
And I think I'll just add that you need to do that risk assessment so that you can identify analytical targets. You won't be able to test for something that you don't know that you don't know what your target is. So these things need to go hand in glove, right? The knowledge you have about your process and product should give you should limit the number of potential nitrosamines that you need to test for. Hopefully, it's a very small number, and you should use that effectively so that you don't have to test, for 1,000 amines, 1,000 potential nitrosamines, and 1,000 APIs. It shouldn't be that broad of a question. You should be able to use the information you have to limit the scope of the potential testing.
Thank you for responding to that question. Our next question is addressed to Dongmei and Deborah and Andre. So we'll allow Dongmei to go first, then pass over to Deborah, and then finally pass over to Andre. And here's the question. How long is the risk assessment, does a risk assessment usually take? Is there a generic checklist created to perform the risk assessment?
The length for the risk assessment, that's really would vary, depends on whether you have a previous experience or some other products, from the same drug manufacturer, whether you have similar root causes before. But by all means, that should be conducted by concluded before March 1st for next year. So currently there is no general template, but according to the guidance, the manufacturers should prioritize the evaluation of the APIs and the drug products based on the factors, prioritization factors such as, maximum daily dose, indication, treatment duration, number of the patients that get treated. And on API side, for the API chemical structure, for the route of synthesis, raw material, intermediate processing aid, any risk factors or, any kind of a nitrosating reagent or precursors you need to think about, and whether that exists in the manufacture process. I will hand over to Deborah and Andre.
Thanks, Dongmei. That's a really good question because I'm sure companies are concerned about how long this is going to take. It's probably going to depend on the process. The simpler the process, the less time a risk assessment might take. And if you go through a lot of the information that the FDA has published, and there's a lot of publicly available information now, a lot of the risk factors are known. We're not back where we were with the sartans, where we weren't really understand how things were forming. A lot of the root causes are now known. And so using that information to take a good look at your process, hopefully that will help you expedite these. But it's going to depend on your process. So I guess that's all I've really got to say, and I'll just hand over.
Yeah, I mean, there's no checklist to do the risk assessment. But one thing I did want to also add to this is that in terms of drug products, and this is something that people probably should start considering, is that we know that in our inactive excipients, they do contain nitrites. That's very well known in the literature. They're quite ubiquitous in excipients. We can see them at hundreds of parts per million levels, even higher and highly variable. So that should be definitely considered in the context of a risk assessment. Drug products have secondary amines. There's that potential for the interaction with the nitrites in excipients. So similar to what is seen in solution phase chemistry that's used to manufacture APIs. So that is something that should definitely be considered in the context of your risk assessment. But again, there's no checklist to do this, but it's all based upon sound scientific principles, and I think a lot of those things were quite well discussed in the slides.
Oh, one more thing I want to add is that FDA may also ask some applicants to consider assessing the risk for some drug products sooner than the others, based on the information we receive and based on our own risk-based evaluation. So in general, FDA will send this request directly to applicants as application-related correspondence.
Thank you for responding to that question. Our next question is addressed to Dongmei. Where can one find FDA-published methods for nitrosamine impurities testing and control?
So FDA already published the method, and for these impurities, actually they're all in the guidance document. If you go into the guidance, and they're all in the footnotes, and actually these all footnotes come from this FDA public webpage. So open the guidance and read it, and you will get all the information for sure.
Thank you for responding to that question. Our next question is addressed to David. Are there methods using the thermo-ion trap method?
Thermo-ion trap method. I'm not sure.
Thank you for responding to that question. Our next question is addressed to Andre. Are all excipients expected to be assessed? Are there some excipients that could be a higher source of concern on the presence of nitrosamines?
Nitrites and excipients, and so there are some excipients that have historically higher levels of nitrites than other excipients. I mean this is I'm not going to give you that specific list, but if you go to the public literature, it's pretty well articulated. I believe a lot of the so I would go to the public literature, and also I would work with your excipient suppliers to see which excipients have high levels of nitrates. That's how I would approach the problem, but not. So I think that hopefully that answers the question.
Thank you for responding to that question. Our next question is addressed to David. Can we develop our method using TOF instrument?
So I guess I think they're referring to a time of flight mass spectrometer. And maybe the earlier question was referring to thermo orbitrap. If that's the case, both those platforms can be used so long as you establish and validate a method with the appropriate sensitivity for your application. So yes, you can use QTOF or orbitrap technology on the mass spectrometry side to do these kinds of measurements. We've also used GCMS-MS approaches, and actually some measurements were made on a triple quad. So there's many different platforms and technologies that are available to do this kinds of testing. Again, it needs to be appropriately validated for the intended purpose.
Thank you for responding to that question. Our next question is addressed to Andre. Should potential nitrosamine contamination be evaluated for products in development? And also it says it should be evaluated in development in the context of risk assessment. And if yes, should this evaluation and risk analysis be included in the NDA?
Yeah, so risk assessment is when people do product development, they do their risk assessment. That's a part, that is part of the product development strategy. So of course, during the product development part, people should do their risk assessments, but one thing I just want to be very clear is that those risk assessments generally don't need to be submitted with the dossier unless, we're seeing a high risk and this is confirmed by testing. If the risk is low, that doesn't really need to be submitted, and it's confirmed by testing that there's no problem. Of course, that probably doesn't need to be submitted to the application in the pharmaceutical development section for the CTD. But if the risk is high, then that should be submitted to the application. To the application. Just bear in mind that if the risk assessment is low, even though you're not submitting to the application, should be retained at the facility for the inspector to look at.
Thank you for responding to that question. We have time for just one more question. We'll try to get that in, and it's addressed to Andre. For pending applications that are close to approval, should we wait for FDA to ask for nitrosamine risk assessment and an IR, or submit the amendment ASAP with the risk assessment?
So it's basically a fundamental question about risk assessment, and I think the most important people need to realize is that whether it's a pending application or it's approved applications, you need to do the risk assessment. And if the risk assessment is deemed to be low and there's no problem, you don't need to submit it to the application, whether it's a pending application or approved application. But if the risk assessment is high and this is confirmed by testing, then yes, you should submit it. So it depends upon the context of the risk. If the risk is low, you don't need to do anything, just keep it at the facility. But the risk is high and confirmed by testing, yes, certainly do amend your application with that information.
That was our last question. We want to thank all of our presenters and panelists for your very informative and timely talk, and also for responding to the questions that came in.